In gel electrophoresis, separated DNA fragments can be visualized with the help of option A, acetocarmine in UV radiation. This is incorrect. Acetocarmine is a stain for visualizing chromosomes, but it is not typically used for DNA fragments in gel electrophoresis. Option B, ethidium bromide in infrared radiation. This is also incorrect. Now, ethidium bromide is used, which is a fluorescent dye that intercalates between the bases of the DNA, but it does not fluoresce under infrared radiation. Rather, it fluoresces under UV light. Option C, acetocarmine in bright blue light. This is also incorrect. First of all, acetocarmine is not used for visualizing DNA in gel electrophoresis, and it does not fluoresce under bright blue light. Option D, ethidium bromide in UV radiation. Now, ethidium bromide, as already stated, it binds to the DNA and it fluoresces when it is exposed to UV radiation, thus allowing for the visualization of the separated DNA fragments in a gel. Hence, it is the correct option.